So yes, let's start with the today's session. Now, as you know that in the last session we have already completed the genetic code and translation, right? Today we are going to learn one of the important concept. Only one concept is, but the concept is little bigger, so that's why just one concept is today. That is called the regulation of gene expressions. So we will focus on that. Let me first of all explain you a little bit about the gene expression. So it will be more easy for you to understand that what exactly this gene regulations and that regulation of gene expression and all, right? So please don't leave the session and join the session in between, so you may forget what I told. So better you be continue, so it will be good for you and me also, right? So as I saw, okay, let me check again. 1 2, okay. Well, so yes, let's start with this. The very first diagram is a reflection over here. What you all understand from this diagram? Can you able to tell me what you are understanding from this diagram? Any idea? Come on, think it. Is this something related to stem cell? And then there is another cell which is going to be controlled by the stem cell? No. Stem cell is not differentiated into various cell, but this diagram exactly what it illustrated. Let me explain you a little bit, so it will be very clear idea for you that what we are going to learn. So actually the diagram which is represented over here, it is, you know, sum of the variability of human cells. Means you can see how many variabilities in the human cell, the shape and other characteristics that make each type of cell unique, right? Like we can say we have blood cell. So blood cell, why we call them blood cell? Because they have unique characteristics, unique shape, variability, right? So you can see here the blood cell, nervous cell, epithelial cells, human cells, sex cells, then there's a muscle cell, fat cells and bone cells. So we have so many cells, so many variations, so many different characteristics, right? So that means of each cell is unique, like, you know, that make each cell unique depends mainly on the specific protein that particularly cell type makes. So proteins are basically, they are encoded in the gene, as you have already studied that in the translation, that how the protein translation take place, right? So proteins are encoded in the gene. Now it is very much clear because we have already studied in the previous session that gene is a particular segment of DNA, right? So that means of, and that DNA when translated, it create the proteins, it form the proteins, right? So protein, in short we can say proteins are encoded in the gene. Means what kind of protein is going to be formed, manufactured, that is in already encoded in the gene. Are you getting my point? Yes. Okay. So what happened? Protein encoded in the gene, all the cell in the organisms have the same gene, right? So they have all gene, you know, genetic instructions for the some proteins, like stem cell have genetic instructions to make this kind of proteins, blood cells have instructions to make this kind of proteins, right? So different type of cell must use a different gene to make a different proteins. Yes or no? So how these different proteins make? Because of the gene regulations. So simply we can say, using a gene to make a protein, using a particular segment of gene to make a protein is called gene expressions, right? It is what? Gene expressions. So as the topic we are studying here, that is regulation of gene expressions. So what is mean of gene expression? You can see the topic is, that is regulation. Let me write here, regulation of gene expression. right? So what is mean of gene expression? As I told you that that the gene that going to what the using a particular gene to make a protein. Okay, so with the help of this gene make a protein, is called gene expression. Clear? Yes, that means of what exactly we are doing, like the gene, by using a particular segment of gene to make a protein. This process is simply called the gene expressions. We will see in details that how exactly it regulated, because regulation is very very important. See, expression gene is very simple, that it is going to be expressed to form a protein. But what are the process? What kind of regulation take place in prokaryote and eukaryote, that we will see in details. So now it is very much clear that what we are going to study today. We are going to study the regulation of gene expressions, and you know very well what is gene expressions. That a gene make the protein is called gene expression. Clear? Now let's move ahead and try to understand. Okay, here is the recall. Now I think so. We have already studied this. Yes. Who is going to explain me? I want anyone from you to explain this. What is, because you have already studied. I have already taught this topic, right? This is what? This is something related to
Translation.
Yes, this is translation, right? So in translation, there are total 3 steps, right? Initiations, elongations, and termination, right?
Yes.
Okay. So you can see here initiation, elongation, and termination. Then what is the product?
Protein.
Protein. So protein in the form of what? It is a polypeptide.
Yes, sir.
Clear? Yes, it is a chain of polypeptides, which we can call it proteins, right? So here you can see, as we have already studied, that in a normal translation process, there is a requirement of large subunit and the small subunit. So you can see here, this is a large subunit, this is a small subunits. So this large subunits and small subunits is going to bind at the specific positions in the mRNA site. So here is the specific positions where it is going to bind. So you can see how they are bind, and there is attachment of anticodon, that is UACTRNA is going to carry out the amino acid over here. So it is going to attach. Now here in this initiation factors, or we can say the larger and smaller subunits, you can see there are total 3 site. What the name of that site I told you? Is it something P site, A site, and E site? Yes, it's APA. Yes. What is the sequence? I want to know the sequence. Suppose this is AUG. So this is PA or E. Tell me what is this sequence? Second number sequence is what? It should be whether it is A or P or E. EPA. That's great. Is it EPA? APE? Okay, now it is very confusing, because N responded APE and Mol responded EPA, but what I feel, EPA is more accurate. Yes, yes, because exit side is here, na? This one. So this is E, this is P, and this is A. So EPA is the
Sir,
accurate
I gave
sequence.
the answer and the process pathway.
Where?
From A side to P side, then E side. I gave that in the pathway, sir, not in the structure.
Okay, fine. But now it is clear for you that first is E side, that is exist side, right? Then there is a P side and A side. Clear?
Yes, sir.
Okay, good. No problem. So initiation, elongations. Now after the protein formations, then there is somewhere there is a stop codon. So once it reaches to the stop codon, translation stop, and all the initiation factors is going to be dissociate, and it release complete polypeptides, right? And the RNA, that means of larger smaller subunit, is going to be dissociate. So this is what we studied in the last sessions and how the protein formation take place. Now today we will go little bit deeper and try to understand about the gene expressions. Now come to the gene expressions. Now as in the initially I have already told you the logic, the idea behind this gene regulations, gene expressions, but let's talk about it. So far now you know all that what is gene expressions. As I told you that gene is the kind of mechanism at the molecular level. So it is, that means of it is just to at the chemical level, molecular level means very minute level. We have to understand that what exactly happening inside the gene, right? So mechanism at which molecular level by which a gene is able to express itself in the phenotype of organisms, means what are the phenotypic characteristics that is going to be control at the genetic, at the molecular level, right? That is what the gene expressions. So the mechanism of gene expressions, basically it involves the biochemical genetics. Means we are going to focus on biochemically how these genes are going to be controlled. So it consists of synthesis of specific RNA, polypeptide, structural protein, proteinaceous, biochemical and enzymes which control the structure and functioning of the specific trait. So as you can see that in the previously we have already studied that how they are going to control. So this is what the DNA, mRNA and protein translation, right? But what are the specific expressions, or we can say the chemicals who basically controlling? So the controlling, you can see the regulation of gene expression by a hormonal label, how it is going to be controlled. So it is biochemical genetics. Now there are two type of gene which basically involve in gene expressions. Now what are those? So very first one, that is called the housekeeping gene. Can you see here the housekeeping gene which is mentioned over here? Housekeeping gene. Now what is this housekeeping gene? So housekeeping gene are one which are continuously expressing themselves in all the cells of the body. Housekeeping gene, you can correlate like this, housekeeper. What is the role of housekeeper? You know very well, right? They have to continuously clean the house, continuously, not continuously, but on the regular interval time, to clean the house, to make the house neat and clean, right? That is a housekeeper. Similarly, here housekeeping gene means what? It is a kind of gene who continuously expressing themselves in all the cell of the body, and because of this particular gene expressions, it is going to be maintained, right? So simply we can say it is what? It is continuously expressing themselves in all the cells of the body, and it is because their product is always required. Why it is continuously working, this gene? Because the byproduct which is after the, it required, always required, there is a requirement of the byproduct, and that's why this particular gene is going to be work continuously. And you can see the example like gene for glycolysis. The best example is gene for glycolysis. Okay, so now it is clear that why it is work continuously. Yes, who is going to tell me that why a housekeeping gene is working continuously? The reason behind? Because the product requirement clear? What kind of product? Like I can say the example like glycolysis. You know glycolysis? Yes, you have already studied in your lower grade in uh, 11th standard, photosynthesis in somewhat or respiration, right? So in that respiration, we've already studied that the glycolysis and what? What is the product of glycolysis? Yes. Okay, breakdown of glucose, but what is the product of glycolysis? Come on, I want answer from you. You have already studied. And this is not just for only one student, this is for every student. Please respond it. Come on. Pyruvic acid, okay. Absolutely. Sarjana, ATP is definitely, it produces ATP, but the product is glycolysis means what? It is simply, the breaking of glucose molecule and conversion glucose to pyruvate. Conversion of glucose to pyruvate is a glycolysis, right? So that means of in a particular cell, pyruvate is required continuously, and that's why this housekeeping gene is continuously expressing themselves in all the cell of the body so that the product is going to be fulfilled. And that's why the based on the requirement, this housekeeping gene is working continuously. Clear? Yes. Anyone have a doubt? See, this chapter, the terminology is very volatile, and that's why I'm just asking you. If you have any doubt, then please ask me. Don't sit and just listen, and you should have to note it down also, because this is something which is very important, and this chapter is simple. If you focus on very carefully, then it is very simple. Yes. So what I said, yes. So anyone have a doubt except Anmol Deep and then Sarjana? Anyone? Because only 3 or 4 students is responding. I don't know why all other are just sitting. Yes, Prasanti, what are you doing? Why not responding? Prasanti, can you hear me? So you have to respond it, right? If you are sitting in the class, that means of you have to respond anyhow. Yes? Sasitaran, you also. Why you off your camera? Please on your front camera. I told you many times that during the live session, during my session especially, you have to on your camera anyhow. Right? Okay. Sasidaran, just tell me, what is housekeeping gene?
So, housekeeping genes, like,
Yes.
they are, like, as every gene constitutes its own thing, like, housekeeping gene are genes which express themselves in, like, everywhere in the organism, everywhere, like, every cell of an organism.
Everywhere or continuously? See, housekeeping gene is continuously working in a particular cell, in, or we can say in the all living organisms. Why it is continuously working? What is the reason behind it? Because the product is required. Like what kind of product? Like suppose there is a requirement of pyruvate. So there is a gene for glycolysis who is going to express continuously so that the product is going to be fulfilled, right?
So,
That's why
like,
this,
continuously they will be expressing themselves in everything.
yes, that is what the main motto, but still you have to just remember like this. See, housekeeping gene, you know, the housekeeper, the role of housekeeper, like, so at regular interval of time, housekeeper, generally they generally keep the house clean and neat every times. Similarly, here also you can just correlate, but you cannot write in theoretical way that housekeeper gene is keeping floor, and then similarly it is kind of housekeeping gene. I'm just giving one analogy, but don't write these things, because when I check the paper, evaluate your papers, many of papers I found that kind of answers. So we are just correlating, we are giving the analogy so to understand you much better. But what you are doing, you're just doing the same things what we are taught, that everything you are writing except the scientific terminology, right? So you should very specific with the scientific terminologies and all, because when writing the paper you should know the scientific word terminology. That is very helpful for you. Yes, so let's focus on. So as I told you, the housekeeping gene, these are the gene which are continuously expressing themselves in the cell of the body because there is a requirement of the product. Like for example, I forgot to write the example. So for example, like gene of glycolysis, gene of glycolysis, clear? Gene of glycolysis. So that can be a good example of housekeeping gene. Now let's move ahead and try to understand the other gene. Now you know very well that housekeeping gene is what. Now there is another gene is also there, and these gene are called the luxury gene. You can see here, these gene are called luxury gene. Why it is called luxury gene? Because these are not expressing their effect all the time. Like this is kind of, you can do work at a time and you can earn money several times. So it is kind of luxurious life. So this kind of gene, they spending their life like luxury. So it is named itself as a luxury gene, right? So let me explain you properly that what exactly this luxury gene. So other gene which are not expressing their effect all the time, they are expressing but not continuously, not all the time, sometimes, right? So these genes are called the luxury gene, right? Luxury gene, or you can call it as a non-consecutive gene also. So it is a non-consecutive gene, or we can say the luxury gene, right? And apart from that, this gene, their activities are regulated, and therefore they are called the regulated gene. That means of this gene's activity is regulated, right? So these are called the regulatory gene. The activity of gene, if regulated, it is called a regulatory gene. So we are very much clear about the housekeeping gene, luxury gene, and a regulatory gene, who regulating the particular phenomena, particular function is going to be regulated. So it is a regulatory gene, right? So the activities which is regulated by particular gene, then it is called the regulatory gene. Now, apart from this regulatory gene, let's focus on another type of gene. We will come on this inducible. See, consecutively gene express, it is nothing but the same like a housekeeping gene only. This is structural gene and inducible gene. We will focus on upcoming slides. Okay, so we just initially we have to focus on this 3-4 type of gene. Now, what is gene regulations? Now we are very much clear about the gene expression, right? Now what is gene regulation? This is important. So gene regulation is what? How we can define the gene regulation? Gene regulation is what? It is nothing but it is a kind of mechanism of switching off and switching on of the gene. Means the gene, when it should be switched off and when it should be switched on, depending upon the requirement of the cell, are called the gene regulation. Means when there is a requirement of the product, suppose in a particular house, if you are studying, then you have to keep your fan light on, right? And when you go out, you have to keep anyhow everything is off. So that means of when you required, you are on. I mean, you are switching on. When not required, switching off. Right? So it is what? It is a regulation. Similarly, so this analogy when we compare with the gene, so gene regulation is what? It is a mechanism of switching off and switching on of the gene depending upon the requirement of the cell, and the state of the development is called the gene regulation. Is this clear? Yes? Okay. Now here the word which is little confusing you, like what is mean of switching off and switching on? Yes or no? Right? Yes or no? Okay, let me explain you. The process of switching on, that means of a gene to produce mRNA and protein, right? Switching on means gene to produce mRNA because it is a requirement of protein. So the mRNA is going to be on and protein formation take place. So it is a switching on, right? Similarly, when the gene are expressed or turning off, or we can say switching off, means there is a no requirement, so it is going to be off, right? That indicate that switching on and switching on is a kind of machineries or mechanisms who managing this, how to at what concentration the protein is required, right? Now gene regulation is further classified into, as you can see here, it is classified into two types. One is called negative and the positive. Negative and the positive. Now what is negative gene regulation and the positive gene regulation? So you have to focus on this positive gene regulation, and because negative in the next slide, so don't focus on this particularly, but you have to focus on this positive gene regulation. Now what is mean of positive gene regulation? Sorry, negative gene regulation. Sorry. So you have to focus on not positive, you have to focus on negative gene, because here is mentioned about the inducible, na? So negative gene. Okay, so in the second diagram you have to focus. Okay, what is second diagram? Negative regulation. Okay, now what is this negative regulation? Negative regulation, gene regulation, that means of the gene continue expressing their effect till their activity is suppressed. What? The gene continuous expressing their effect till their activity is going to be suppressed. That is what called the negative. Okay, as the gene express their effect through the enzymes, and their enzymes are called the inducible enzymes and the repressible enzymes. Okay, so now it is clear what is negative gene regulation. Those the gene which continue expressing their effect till their activity is suppressed, right? In the negative gene, expression is called the repressible regulation, right? This in the negative gene regulation, this negative regulation is called, it's a repressible regulation. It is called repressible regulation. Okay, and repression is due to the product of regulatory gene. Why there is a repression? Because it is the product of regulatory gene. Regulatory gene. Clear? So it is a negative regulation. Similarly, in positive regulation, what is positive regulation? Okay, so we have to focus on this only. Now focus on the second diagram, first diagram. Positive regulations. What is positive regulation? Again, positive regulation is the one in which gene remain non-expressed unless and until they are induced to do it. Means there is a requirement of inducer, right? So if there is a requirement of inducer, then only this gene is going to be expressed. So it is called positive regulation. Okay, is the difference is clear to both to all of you? Yes.
Sir, in the second diagram, the repressor molecule is actually the inducible enzymes or repressible enzymes, sir.
In the second diagram.
In negative
Okay.
regulation.
Okay. See, in the negative regulation, that means of the gene which is continuously, see what is actually happening in this particular negative gene regulation. The gene continue expressing their effect till their activity is suppressed. So that means of it continuously expressing until it reaches to the suppression stage, right? So negative gene expression is also called the repressible regulation because the repression is due to the product of regulatory gene. But in case of positive gene regulation is the one in which enzymes remain non-express, means it is still sitting at one place, no any activity. When till someone is going to come and induce it, come on, do it. So it is going to start doing that work. So that is called the positive gene regulation. So you can just focus on here, like here in this diagram. See, this is a activator. Activator means what? That some inducer is there. So when the some inducer is going to bind with this, it is going to be transcriptions. That means of it going to transcribe. If there is a no inducer, means it is there is a no transcriptions. Clear? This is what called positive gene regulation. But in the negative gene regulation, you can see negative gene regulation is continuously expressed in the second diagram. It is expressing continuously, right? But when it expressing continuously till there is a suppressor, till it is going to be reach up to the suppressor. So here you can see there is a repressor who is going to stop the reactions. And once the repressor bind to this, it is going to stop, no further regulation take place. So it is a repressible regulation gene, we can say. Why? Because there is a repressor is required to stop the reactions. Is that clear to everyone about what is positive gene regulation, what is negative gene regulation, and any doubt in this? And
No, sir. So in positive regulation, something has to induce so that the transcription takes place.
yes,
And in negative regulation, if something is suppressed, transcription will not take place.
not take place. No, that means something is repress. Repress means it is going
Something is going to inhibit or block it.
Yes. So that's why and when it's blocked, no transcriptions take place, right? So it is a negative gene regulation. Clear?
Yes, sir, clear.
Good. Any other have a doubt? So you can just ask me, because this gene regulation is little difficult, but I'm trying my best to at least to understand you what exactly this. So I'm sure that everyone is very sure about that. They are clear all these things. But still, if you have doubt, then please ask me. Now here Kirtini, Sarjana, then Prasanti, Anmoldeep, Akhilesh, Sidaran. So number is used, but no one responding except Anmol, Sarjanan, Prasanti, and one more Ann. Only four students responding in this class, and rest of all just sitting and watching. We are not just here like a movie. We are just speaking, and you are just listening. You have to respond also. You have to take active participation in the live session, right? See, let me tell you one thing very clear. If you are actively participating in the community, I mean to say in the interaction, then only it is easy for you to understand the concept. If you are sitting and not doing any kind of activity, then it is, you know, like that your understanding is very we can say it's a zero level, because you're not participating in the what we are talking. At least you can say yes, no, got it, yes, not. At least that word you can. I think so. This is very simple word, right? You have to just use that chat box and tell me that whether you are understanding or not. See, Sasidharan not responding. Why? Still he is in the class. Akhilesh not responding. Still he is in the class. Anmoldeep responded, no problem. Prasanti, okay, Prasanti responded. Kirthini just now responded. Gopinath, no response. Why? Okay. Yes, got it. Sasidharan, no problem. Okay, shall I move ahead? If this concept is clear to you, positive gene regulation and a negative gene regulation, so can I move ahead? Okay. Good. Now, let me tell you something more about it. Now, this is very clear about the positive and negative gene regulations. Now, as there is a requirement of some, inducer, and therefore it is called inducible regulation. What? As you can see here in the positive gene regulation, there is a requirement of inducer. This is inducer, right? Inducer. So it is called inducer gene regulation. As like here repressor gene regulation, similarly there is a inducer gene regulations. So here it is inducing and here it is repress. So it's a repressible gene regulation. It is inducer gene regulations. Now is this clear? What is inducer gene regulation? What is repressible gene regulation? Okay. Now come to the next, that is gene regulation in prokaryote and eukaryote. So gene regulation in eukaryote is little complicated, and it take place at 4 level. What are those 4 level? The first level, that is called the transcriptional level, means formation of primary transcript. As you can see, first of all, suppose that this is the particular segment of gene on the DNA. So the first is a primary transcript. Then processing label means regulation of splicings. So this is the processing label. Then transcriptions of mRNA from nucleus to the cytoplasm. Transport of mRNA means once the mRNA, you know that in the eukaryote. Okay, let me one more thing. In the last session, I've also told you that about in which place I mean to say translations take place in prokaryote and eukaryote at different organelles, right? I have told you, right? If I'm not wrong. Yes? Yes. Okay. So in prokaryote, it take place in the cytoplasm. Why? Why in the cytoplasm? You know that in the prokaryotic organism are single-celled organism that lack a cell nucleus. If in that particular cell there is no nucleus, that means of their DNA floats freely in the cell cytoplasm. And when particular protein is needed, the gene that code for it transcribed in the mRNA, which is continuously translated into proteins, right? But in case of eukaryotic cell, in contrast, we can say the intracellular organelle that we know that is called the nucleus, right? So DNA is contained inside the cell nucleus where it is transcribed into mRNA, and that mRNA are supposed to be transported to the cytoplasm, a newly synthesized mRNA that need to be modified, then it modified and transported out of the nucleus into the cytoplasm. Yes? Is this clear what I mean to say? Yes, that means of suppose here, suppose this is cell, okay. Inside the cell there is nucleus, okay, and inside this nucleus there is a formation of mRNA. So this is just consider this is mRNA. Now this mRNA is supposed to transport outside, means it is going to modify and transported outside into the cytoplasm. So now it is transported in the outside. So this is what? This is cytoplasm, and this is nucleus. Clear? So transportation of mRNA from nucleus to the cytoplasm, you know why? Because the ribosome present outside the nucleus, right? So that's why it has to transport from the nucleus to cytoplasm. And then followed by there is a translational label. That means of gene regulation in eukaryote, it is take place by 4 important steps. The one is transcriptional label, processing label, transport of mRNA to the nucleus to the from the nucleus to the cytoplasm, and the translational label. So there is 4 label. But in case of eukaryote, gene regulation, in prokaryote, gene expression is regulated mostly at the site of the transcriptional initiations. Now I have told you that why it is at the site of initiation, because there is no nucleus, so it is suspended in the freely moving in the cytoplasm. So these regulatory proteins can act both activator as well as repressor. Means one regulatory proteins, they are going to perform both kind of action. One is called regulatory and the repressors. Functioning of operator depends upon the protein product, that what kind of protein product is required, that is depends, that is called the operator, that basically depends on operator. Clear? So that is what the logic behind. So it is, now it is very much clear about that, which in the prokaryote and eukaryote how the gene expression take place. Clear? Now come to the our important concept, and that concept is called, okay, another is regulation of gene expression referred to the controlling and functioning of gene, okay, that is environmental. Now come to our important concept. Based on what we studied till now, we have just studied the terminologies of what is repressor, what is negative gene, what is positive gene regulation, what is regulatory gene, what is repressor, inducible, and all. This is all the terminology what we studied till now. Now actual concept started from here. Okay, what is this? This is called the Operon concept. Okay, now what is this Operon concept? So let me tell you, Operon concept was, from the number of studies on the metabolism of bacteria. There are n number of metabolic activities, means how the particular bacteria is metabolized. Okay, so based on the study on metabolism of bacterium, Escherichia coli, Escherichia coli is a kind of bacteria. Simply it is referred as a E. coli. E. coli. Here you can see Escherichia coli, right? So E. coli bacteria, the two French scientists, the name is Jacob and another one is called Monod. So here is Jacob and Monod. One is geneticist and another one is a biochemist who worked together, and they proposed in the year of 1961, they proposed a model of gene regulation, and that model of gene regulation is called the Operon model. Okay, that is what called Operon model. Clear? Now let's focus on this Operon model. Operon is what? It is a coordinated group of gene. Operon is what? It is a coordinated group of gene. Operon means what? It is coordinated, a group of gene. Okay, it is coordinated, a group of gene is an operon. Okay, now what type of gene are coordinated? That is also important, right? So there is a structural gene, there is an operator gene, there is a promoter gene, a regulator gene, which function together and regulate a metabolic pathway as a unit, is called coordinated group of gene. So you can see here in this diagram, can you able to find, I think so in the previous diagram, yes, here you can see this is the regulator gene, operator gene, promoter gene, structural gene, right? All these genes are required to regulate the proper operons, and this is what the Operon model. Clear? Okay, so if anybody asks you what is Operon, Operon is what? It is nothing but it is a coordinated group of gene which basically regulate, which help in the gene regulations, and these genes are includes, what are the gene? That genes includes what I said about that gene, that is regulator gene, promoter, operator, then structural gene. All these are the gene which are continuously expressing. Clear? Now let's try to understand one by one that what is this structural gene, what is operator gene, and all we'll see in the details. So very first you can see the structural gene, right? Structural gene. Now what is structural gene? Simply structural gene is what? That it is transcribe the mRNA from polypeptide synthesis is called structural gene. That means of when mRNA transcribe to form a polypeptide synthesis is called structural gene, right? Similarly, there is a promoter gene. It is a gene which receive the product of regulator gene. Okay, it is a gene which receive the product of regulator gene. Apart from that, it allow the functioning of operon, means because it's operator now, so it allow how to functions. Okay, so it allow the functioning of operon when it is not covered by the biochemical produced by the gene regulator gene. Clear? Similarly, there is a promoter gene. Promoter gene provide attachment site for RNA polymerase. Then there is a regulator gene. It synthesize a biochemical or the regulator proteins which can act positively as a activator and negatively as a repressor. So now we know very well positively activator and negatively repressor means what, right? So they are nothing but they're the regulator gene. Clear? So these are the four important gene which is responsible. So all these four gene working together, that is what called the Operon. And Operon is what? It is a coordinated group of gene, basically included structural gene, operator gene, promoter gene, and regulator gene, who function together and regulate the metabolic pathway as a unit. That is what called Operon. Is this clear? Okay. Now, apart from that structure, Operon gene, there is a repressor and inducer. Now you know very well repressor and inducer meaning is what, because we've already studied the negative in the negative and positive gene regulation. So repressor and inducer, or we can say the co-repressor from outside, are also found, like operator, promoter, regulator gene constitute the regulatory gene. Operon system are common in prokaryote, right? So as you can see here, from outside means what? Repressor and inducers or co-repressor. Co-repressor means apart from this repressor inducer, there is some other outside gene can also involve in that. So they are the outsider genes. So we can say it is a co-repressor gene are also found in that Operon concept, in that Operon model. So operator, promoter, and regulator gene constitute the regulatory gene, right? So Operon are of two types, but there are various types because various scientists work behind. But basically, this Operon is of two types. One is called the Lac Operon, which is called as an inducible, and the another one that is called the Tryptophan Operon, which is referred as a repressible. Means Lac Operon is inducible because there is a requirement of inducer. That's why it is an inducible. Another one is a tryptophan. Okay, tryptophan Operon, which is called the repressor. Why? Because there is a requirement of repressor to stop the reactions or stop transcriptions, right? So there is a Lac Operon and Tryptophan, and these two Operon is very important for your NEET. So this you have to focus on this. This is little important. Okay. Now let's move ahead and try to understand this Lac Operon and remaining. So very first, Lac Operon, which is also called an inducible Operon system, and you know the meaning of inducible because some inducer is required for the reactions, right? So very first, Lac means what? Lac is something. It is also called as a lactose. Okay, Lac means what? It is a lactose, or you can refer to lactose. So in E. coli, now tell me, E. coli is a prokaryote or eukaryote? E. coli bacteria is a prokaryote or eukaryote? Prokaryote. That means we are studying a prokaryote Lac Operon. Okay. Now let's focus on this Lac Operon in especially E. coli bacteria. The breakdown of lactose, because we are studying Lac Operon, right? So the breakdown of lactose require 3 enzymes. Okay, that means in a particular plate there is E. coli bacteria, and there is some amount of lactose in that plate. So bacteria require 3 different kind of enzymes to break the lactose into glucose, right? So these enzymes are synthesized together in a coordinated manner by the functional unit of DNA. Means which DNA? The DNA which present inside the bacteria, right? That is what the Lac Operon. So since the addition of lactose itself stimulates the production of required enzymes, thus it is called the inducible Operon system, right? Now it is clear why this Lac Operon is called inducible Operon system. Why? Tell me why. What is my question? My question is why they require, why it is called the inducible Operon system? Because the addition of lactose itself stimulate the production of required enzymes, right? So that's why it is an inducible Operon system. Like suppose, let's take one simple example. Suppose in your house, uh, on your breakfast table, you have a simple food like we can say idli and dosa, okay, and in another side there is some kind of burgers. So which you prefer first, burger or idli dosa? Tell me. Dosa, okay, and very cultural, okay. Say simple, yes, I need dosa, no problem. Burger. See, I know this answer from only Sanjana, that Sarjana, that she. Okay, why burger? You know, because burger is inducer. Podi dosa. What is this podi dosa? Podi dosa. What is this? Prasanthi said and not responding. This is a kind of side dish for dosa, sir. Okay, it's a chili pot. Okay, stuffed inside the dosa. Okay, this is something unique. Podi dosa. Okay, I heard first time this. Well, so my basic message to convey about this idli dosa and burger, because burger is more inducible because it is something new, right? And something new means definitely you will first want to taste whether this what is the taste of the burger, right? Because idli dosa is a general, and you it is always we can say that it's a common generally made every day in our breakfast. But burger is sometimes, you know, it is very unique, and it will so definite, that means a burger is work as an inducer. So similarly, once there is lactose, bacterial gene going to in at the DNA level is going to because of that lactose it is going to be induced. So simply the basic meaning of what I said, that is, it is going to be actually it stimulate the production of required enzymes, and that's why it is a inducible Operon system. Now it consists of this Lac Operon is consist of the following structures. What are these following structures? The very first one is called structural gene. Okay. Yes. Yes, Prasanthi, now focus. Don't focus on podi dosa. Now you have to focus on this Lac Operon system. Okay. So the very first part is structural gene. This structural gene are further or of three types. Three structural genes are present in that. The first one is a Lac Z, Lac Y, and Lac A. So you can see Lac A, Lac Z, Lac Y, and Lac A. Now what is this? The very first one is Lac Z, right? So Lac Z code for, now why it is having some kind of codes, right? Lac Z, why Z is there? Because the Z gene is code for beta galactosidase. Z gene is code for beta galactosidase. Okay, so you have to please note it down. Lac Z, Z means it is code for beta galactoside. Now this beta galactoside means what now? Because which is a primarily responsible for hydrolysis of disaccharide. That means of lactose into its monomeric units, that is called galactose and glucose. Means because of this beta galactoside, lactose, let me write here somewhere, yes I can write here that lactose is going to break into galactose and the glucose. Galactose and glucose. Clear? What is Lac Z? Yes. So now it is clear that Lac Z is code for beta galactoside, which having responsible for hydrolysis of disaccharide into a monosaccharide. So actually lactose is a disaccharide which is going to convert into a monosaccharide or monomeric unit, that is called the galactose and glucose. Clear? This is the function of Lac Z. Similarly, come to the Lac Y. Now this Lac Y gene code for permease. Okay, it is code for permease. Okay, now what is the function of this permease? So basically, which means it increase the permeability of the cell to the beta galactoside. So basically it increase the permeability. Okay, so it increase the permeability of the cell. So for what? To beta galactosidase. And the last one is Lac A. So Lac A is code for trans-acetylase, which can transfer the acetyl group to the beta galactoside. So it is going to transfer. So basically it is a trans-acetylase. So that means of it is going to transfer acetyl group. Okay, it is going to transfer acetyl group to beta galactoside. Is clear? So this is 3 important structural gene. Is this clear to everyone? Oh, sorry. Okay, now the next. So after structural gene, the next is operator gene. So operator gene is what? It interact with the protein molecule or the regulator molecule. Basically operator, so it basically interact means it going to operate in between the protein molecules and regulatory molecules. Then there is a promoter, the gene which poses site for the RNA polymerase attachment, and definitely there is a regulator gene, that is the gene which code for protein known as a repressor protein. So these are 4 important gene which is responsible for Lac Operon and their functions together. Now let's try to understand when there is so very first Lac Operon Inducible system when it is off. Okay, that we need to understand. So very first, now you can see here, yes, so regulation of Lac Operon by repressor is refer as a negative control or negative regulations, because we have already studied what is negative control. Means where there is a repressor who is going to suppress, right? So it is a repressor. So the regulation of Lac Operon by repressor is referred to as a negative control or regulations. Clear? Okay, so when it is switched off, that is my question, right? So I think so the diagram is not accurate over here. Operator in structural gene bind repressor. Okay, repressor is there, yes. So now let's try to understand when it is switched off. So very first, the operon is switched off when a repressor protein produced by the regulator. So you can see here, this is the repressor protein, right? And this repressor protein is produced by whom? By the regulator gene. So you can see this is the regulator gene. This regulator gene, when produce the repressor gene, what happened? Which bind to the operator gene. That means of this gene is repressor gene going to bind to the operator gene. And when it bind to the operator gene, what will happen? You can see this operator gene, the RNA polymerase get blocked. So here what happened, once it bind repressor, the RNA polymerase blocked. Why blocked? Because this repressor bind on the operator gene. So when this repressor bind on the operator gene, RNA polymerase blocked, and once RNA polymerase get blocked, so there would be no transcriptions. As you can see, can you see there is a transcriptions? No. If no transcriptions, that means of this is what called the switched off. Clear? This is what switched off, because repressor bind to operator and operator block the RNA polymerase, and then there is no any further process. So it basically it is a switched off. Now what is in case of switched on? Now what is switched on? Let's try to understand. So in case of switched on, yes, in case of switched on, now you can see if lactose is provided in the growth media, that means of here in the initial one there is no any inducer, but here that means of in the previous can you able to find any lactose over here? No, because here is no any inducer, but in the next you can see there is an inducer. Okay, what is the inducer? In the form of lactose. That means of in that bacterial plate, when there is a media contain lactose, it is work as an inducer, and because of that inducer, the lactose is transported into the cell through the action of permease. You know that that lactose, because of three important enzymes I have told you in the starting, right? So very low level of expression of Lac Operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cell. So in the presence of an inducer such as lactose, or we can say the allolactose, the repressor is inactivated by the interaction of the inducer. So you can see once there is a lactose present, this repressor is going to become inactivate, right? Because in this media there is a lactose present. If there is a lactose present, so this repressor become inactive. Why? Because there is a lactose, so it become inactive, means there is no any kind of activity. So if it is not active, means it is not going to bind on the promoter gene. Yes or no? Come on, respond it. If it is not, so it is not going to bind on the promoter gene, right? So if it is not going to bind on the promoter gene, that means of the repressor is inactivated by the interaction with the inducer, and these allow RNA polymerase access. Means once it
Sir,
is not, yes, please.
it won't bind on operator side, no, sir.
See, what is my explanation over here? I told you that this repressor is not going to active. Why? Because there is a lactose present. Clear? So if it is not bind, this repressor if not going to bind on the operator gene, then what happen? If it is not binding, so here is a RNA polymerization take place continuously, because RNA polymerase is not going to bind, I mean to not going to block. Yes or no? Because see, in the previous case, just correlate with the previous case. Here what is happening? Repressor going to bind to the operator, and because of it bind to operator, RNA polymerase blocked. Why? Because here is the absence of inducer. Absence of inducer means what? No glucose, no lactose. No lactose. Clear? But in case of the inducer switched on condition, what happened? This mRNA, definitely there is a regulatory gene who is going to produce the mRNA, and this mRNA produces the repressor, but repressor is going to become inactive. Why it is inactive? Because of the presence of lactose. So once this lactose is present, this is not going to perform any kind of functions, means it is not going to bind on the promoter gene, and once it is not bind to the promoter gene, that means of RNA polymerase continue. And if it is continue, then what happen? It allow RNA polymerase to access the promoter and transcription proceed, and that's why this is what? This is the condition which is called the switched on, right? Means if it is switched on, so what happened? There is a lac mRNA. So this lac mRNA, beta galactosidase, permease, and transilase. So this lactose is going to with the help of beta galactoside, it is converted into glucose and galactose, because of lactose is present. So this lactose is going to break down, because of this enzyme it is going to break down into glucose and galactose. So glucose and galactose is what? It is a monomeric, yes, and lactose is a dimeric. Are you getting my point? Yes. So this two important. When the Lac Operon is on and off, that you should know. If there is presence of lactose, what happen? It is switched on or switched off? My first question. If in the particular plate there is a bacteria, and in that bacteria there is no lactose, what will happen? Off. Very good. Very good. See, again I can see only 3 and 4 students only responding. Very bad. I don't know why. For me, or I cannot say anything. But at least you should have to realize why you're not participating in that chat or in that discussion. Okay, just tell me, is my topic is boring?
Sir,
Yes.
sir, can you please tell that in switching on, there is an inducer that is lactose.
Hmm.
Is it bind with is it bind with operator to
No. Let me tell you. Anmol Deep. See, it's very simple. Just a minute. That is, in the case of switched on. Okay. So in the case of switched on, This is the regulator gene. This regulator gene produce repressor mRNA. Repressor mRNA, that means of repressor gene. But this repressor gene is going to become inactive because there is a presence of lactose. So if it is inactive, then it is not going to bind to the promoter gene. And if it is not going to bind the promoter gene, then there is a RNA polymerase who is going to be continue. So it produces the Lac mRNA, and this Lac mRNA with the help of three enzymes, that is beta galactosidase, permease, and transacetylase. I have told you that what is this all, right? And so basically Lac permease, its permeability, transamylase because it transfer the acetyl group to beta galactoside, and the beta galactoside which convert lactose into glucose and galactose. Clear? So this is what the switched on condition. But in case of switched off condition, what is happening? Regulator gene produce repressor. This repressor going to bind to the operator, and once it bind to operator, there is a blocked of mRNA polymerase. If there is a blocking of mRNA, then there is no mRNA is going to produce, and if no mRNA, that means of no further reaction will take place. That means of no lactose is going to convert into glucose and galactose because there is absence of lactose. That's why it becomes switched off. Here, Anmoldeep. Okay. So we studied about the Lac Operon. Now there is another Operon which is called the Tryptophan Operon. Now what is this Tryptophan Operon? Let's try to understand. Now here in the case of Tryptophan Operon, bacteria like Escherichia coli need amino acid to survive. Why? Because they need to build a protein, and one of the amino acid they need is tryptophan. Clear? So that's why it is named as a Tryptophan Operon. So let's try to understand in details about it, this Tryptophan Operon. So basically this Operon model can also be explained using the feedback regulations. So in the Tryptophan Operon, three enzymes are necessary for the synthesis of amino acid tryptophan, and these enzymes are like 5 different coordinated manner. Like what are these 5 different coordinating manner? So the addition of tryptophan stop the production of these enzymes, and thus the system is known as a repressible system. So that means of tryptophan is a repressible operon, and Lac Operon is inducer operon. This is a repressor, and the another one is an inducer. So let's focus on this. So if the tryptophan is present in the environment, that means of suppose in a bacterial plate there is a tryptophan. In the case of no lactose, no any other things, but there is a tryptophan. So then E. coli bacteria don't need to synthesize it. So the transcription of gene is TRP operon is switched off because it is already present. If tryptophan is present, so there is no need to synthesize it. If you have already had a food, so no need to extra food requirement, right? Because you are already full. Yes or no? Is it similar kind of that if the tryptophan is present in the bacterial environment, don't need, so that means of this bacteria don't need to synthesize it. So transcription of gene in the TRP is switched off. In the another case, when the tryptophan availability is low, that means of in that environment tryptophan is utilized and it is going to be low in number, or we can say low in quantity. So on that case, on the other hand, the operon is switched on and the gene are transcribed and biosynthesis enzymes are made and more tryptophan are produced. That means of whenever you require you can produce, whenever not required you cannot. So not if you cannot require, so it is switched off. When you required, it is switched on. It's like similarly when you hungry, you have to eat lot of food. That means of your system is on. When you're not hungry, you have to be sit silent because you are full, right? So it is similar that kind of in tryptophan, because tryptophan is available in that environment, so bacteria is not going to synthesize it, so it is switched off. If it is synthesized, if it is going to be low down, so it is going to be synthesized again. This is what the tryptophan operon. Is this clear? Now here, so switched on and switched off is clear. Now the TRP operon includes 5 genes that encode enzymes needed for the tryptophan biosynthesis, along with the promoter and operator. So the gene of tryptophan are transcribed as a single mRNA or either single mRNA, or either it can be a with and that 5 tryptophan 5 structural gene are, let me write that 5 structural gene. Yes, it required tryptophan having 5 gene, and that gene is, it is referred as a E, A, C, B and A. This is the code. Okay, so tryptophan includes 5 enzymes that encode enzymes needed for tryptophan biosynthesis, and the name of that enzyme is enzyme E, enzyme A, enzyme C, enzyme B, enzyme A. Okay, sorry, E A E D A C. This is slightly wrong. This is D. So E D C B A. This is what the enzymes. Okay. Okay. Now come to the turning off and turning on. Okay. So turning off and turning on. So this stretch of DNA is recognized by the regulatory protein known as a TRP repressor. Now what is TRP repressor? So you can see high tryptophan. If it is high tryptophan present, RNA tryptophan is present, then what happen? No transcription take place. You know very well why no transcription take place. As I told you. Na? Yes or no? If the tryptophan is high in amount, definitely it is not going to be transcribed, and that's why no transcription take place. These are the enzymes is going to be inactive because no tryptophan required. So when the repressor bind to the DNA of the operator, it keep the operon from being transcribed by physically getting in the way of RNA polymerase and the transcription enzymes. But in case of, and when there is a little tryptophan is in the cell, on the other hand, the TRP repressor is inactive and does not attach to the DNA or block the transcriptions, and this allow the TRP operon to be transcribed by the RNA polymerase. Means in the second case, you can see this is, suppose this is a low tryptophan. If it is low tryptophan, then it is going to bind, and if it is bind, then the transcription will take place and there is a synthesis of tryptophan in a large amount. So it sends whether the tryptophan is already present at high level, or if so, it switch the operon to off positions, preventing unnecessary biosynthetic enzymes from being made. That is what the turning operon on and off, and that is related to operon if it is low and high based on that. Okay, is this clear now? Okay, now here you can see the regulation of gene expression in eukaryote. So the regulation of gene operon in eukaryote, so it was, you know, the Davidson gene battery model, okay, is the most popular. It is written already. Yes, it is Britain Davison's gene battery model. This is a kind of model, so you have to remember this name of the model. Okay, is the most popular for the eukaryotic gene expression. It is very much popular. Okay, now it proposed the occurrences of 5 type of gene, and that 5 type of genes are producer, receptor, integrator, sensor, and enhancer, silencer. So you can see here the one by one. That is, first one is a producer or structural gene. So this gene transcribed only mRNA. Similarly, there is a receptor or operator gene. So you can see this is a receptor over here. So this is a cell membrane. This is receptor over here. So every producer gene seems to have its own receptor or the operator gene that lies nearby. So this is receptor gene. Then there is an integration or the regulator gene. So it controls the functioning of operator gene by performing an activator RNA, and this RNA regulates the functioning of receptor gene. So it basically regulates the functioning, how exactly. And then the fourth one, that is called the sensor gene, whose sensing, that gene is meant for picking up the information of the intracellular environment. So it's sensing that particular information, and it. And the fifth one is enhancer silencer gene. So enhancer silencer, these genes are the rate of transcriptions of the structural gene. That is what enhancer. See, enhancer and silencer means it is going to enhance and to become a. So it is enhancer silencer gene. Now, so as of now, we studied about the gene regulation in prokaryote and gene regulation in eukaryote. Okay. Now, yes, anyone have a doubt? Okay. Now let's focus on significance of gene regulations. What is the most important significance of gene regulation? So advantage of gene regulation, that a number of regulated gene required for particular metabolic activity can be switched off or on simultaneously. Means whenever there is a requirement, it is going to be switched on. When there is no requirement, it is going to be switched off. So the very first, it is related to number of gene. Now, gene regulation enables the cell to adjust metabolisms as per the requirement of environmental and developments. Like suppose your body required energy, so that means of your gene is going to be regulate accordingly, right? And in the fourth point, it economically, as it is synthesized enzymes only when there is a requirement. Means if there is a requirement, it is going to be synthesized. Means it is very economical, you know, right? Like because suppose there is no use of light in your bedroom, that means of what? Or we can say in your house, that means of if there is no use, means you are using that light in a very economical way, because you whenever required, you are owning that light. I mean to say switch on that light, and where no requirement, you switch off that light. Means you are using that light very economically or very carefully. So that is what it is economically, as it is synthesized enzymes when there is a requirement. Another one, gene regulation helps in growth and differentiations. It also helps in the smooth completion of chain reactions, right? So this is what about the significance, total 5 important significance. Apart from that, let's try to understand the difference between the inducible and repressible operon. So what are the major difference? So you can see here the differences. In inducible operon, it is an operon which normally remains switched off, and it is switched on by the presence of biochemical called inducer. So when there is an inducer, then only it is going to be switched on, otherwise it is switched off. Induction of operon involves removal of repressor from the operator gene. Regulatory gene of operon produce a complete repressor, and induction start transcriptions translation. Inducible operon is functional in the metabolic pathway. In case of induction is caused by the new metabolites. So these are the regarding the inducible. Similarly repressible, so it is opposite. So it is the operon which normally remains switched on when there is a inducer. So no inducer is required, so it is called repressible operon. And repressor involve the binding of repressor over the operator gene. The regulatory gene form only a part of repressor. Always keep in mind, the regulator gene only going to form the repressor. Now we have already studied that how the repressor gene come. So it is from the regulator gene. Repressor stop transcriptions and translation, and it connected with the anabolic pathway. So this is the catabolic pathway, this is anabolic pathway. This is very important, catabolic and anabolic pathway. And here it is, repressor is caused by the excess availability of existing metabolites. Means if there is excess amount like tryptophan is there in a huge amount, it is not going to be synthesized. So it remains switched off, right? So this is what the major differences between the inducible and the repressible operon. Now let's try to solve few questions based on this topic. Now here the question is, okay, before these questions, let me ask you again, anyone have a doubt? Yes. Anyone have a doubt, then please put your question in the chat box. If you cannot able to ask the question, so you can keep your question in the chat box so I can read and I can tell you the answer at least. Otherwise, I want response from everyone that no doubt in the chat box. Okay. Let's move ahead now and try to solve these questions. Which one of the following is not a part of transcription unit in the DNA? Okay. Yes. So which of the following is not a part of transcription unit in the DNA? So there is a terminator, promoter and structural gene, but inducer gene is not a part of, right? So promoter, structural and terminator is a part of transcription unit. Inducer is a kind of lactose, which is not a component of transcription unit. So the right answer is A, but in here it is written as C, it should be A. Clear? Another one. The sequence of the structural gene in the Lac Operon is. What is the sequence? Sequence is very important. And I have told you that sequence. Yes, it's a Lac Z, Lac Y and Lac A. Another one. Which of the following is required as an inducer for the expression of Lac Operon? Inducer. Whether it is galactose, lactose, galactose and lactose, glucose. I think so this is very simple, like burger. No, galactose, lactose is what? Burger. Can we say like that? Yes. So it is a lactose. Well, now I think so one more question is there. The structural gene of Lac Operon transcribe mRNA, which is polycystonic, replicative, monocystonic, or monokaryotic. So it's a polycystronic. Very good. Oh, yes. Now this is what we studied. So we studied about the Lac Operon regulation in the absence of inducer, in the presence of inducer. We also studied about the inducible operon negative regulation by expression. So inducer, this is a lactose, it should be lactose, not a fructose. It consists of I gene. I gene means regulatory gene. So I gene means, don't forget, I gene means what? It is a regulatory gene which is in code for the repressor, which act as the inhibitors. Then there is a Lac Z, which is code for beta galactoside, and this beta galactoside convert lactose into galactose and glucose. Then there is a Y gene, which code for permease. You know, the permease, it increase the permeability of the cell to the beta galactoside. And then there is, this is A gene, so code for trans-acetylase. So trans-acetylase metabolize the toxic thioacetamides, which are also allowed to enter the cell of the permease. So this is total 5 type of gene: I gene, Z gene, Y gene, and A gene. Now this is the complete topic.